Now we will see about the rabies virus infection. So the first thing to know about the rabies virus is that the rabies virus is a enveloped RNA virus. It's an enveloped RNA virus. And what is the shape of the virus? It has a characteristic shape which is called as the bullet shaped. So it is a virus which is having bullet shape. And regarding the strains of the virus, we have got two strains of the virus, that is the street virus strain and the fixed virus strain. The street virus strain means those strains of the rabies virus which are obtained from the street animals which are infected with the rabies virus, while the fixed virus strain are those strains which are which are obtained in the laboratory, and after passing the virus from one animal to other animal, like from one brain of one animal to uh, other animal, passing it multiple times from brain to brain of different animals. The strain obtained finally is called as the fixed virus strain. Now there are certain very important differences between these street viruses and the fixed viruses. So the difference between the street and the fixed virus is that in the street virus we see that the inclusion bodies are present, while in case of fixed virus strain the inclusion bodies are absent. This is very this can be remembered very easily. See here it is written FIX. IX means I means inclusion bodies, and this cross sign means not present. That means in fixed virus strains inclusion bodies are absent. We cannot demonstrate the inclusion bodies in the fixed virus strains. The other point of differences are that in straight viruses can infect the salivary gland, while fixed viruses do not infect the salivary gland. And in case of street virus the incubation period is long, while in fixed viruses the incubation period is short. Okay, in fixed viruses the incubation period is short. So these are the point of differences between the street viruses and the fixed virus strain. Now coming to the pathogenesis part. So in pathogenesis we have to first see what are the sources of infection of the rabies virus. The most important source of the rabies virus infection are the animals which are infected with the rabies virus, like dog, bat, etc. So by the bite of these animals which are infected with the rabies virus, the rabies virus infection may occur. Other than that, there are some other methods of spread of the virus, that is the direct contact of the saliva. That is the direct contact of the saliva of the infected animal to any wound site or to any site where there is discontinuity in the skin. So if there is discontinuity in the skin and over that uh, uh, wound or trauma site, if animal infected with the rabies virus licks on that, then there may be spread of that rabies virus to that person. Other than that, it may also spread by the inhalational route of by inhalation of the aerosols. Other than that, it can also spread by the corneal transplant. It can also spread by the corneal transplant. So these are certain methods by which the rabies virus may get transmitted. So now coming to the basic pathogenesis of the rabies virus, what happens when the animals which are infected with the rabies virus bites to any human being, they the viruses just simply get inoculated at the site of bite with the saliva of the animal. Now, after inoculation, these viruses multiply locally in the muscle or at the site of the bite. And next, the viruses, as after multiplication, the number of the viruses increases, these viruses enter into the peripheral neurons. So, in the nerve endings of the peripheral neurons, these viruses enter, and via the retrograde axonal transport, they reach to the dorsal root ganglion of the spinal cord. They reach to the dorsal root ganglion of the spinal cord. From the dorsal root ganglion of spinal cord, they reach to the CNS. Through that spinal cord tract, they reach to the CNS, and thereby the manifestations of the rabies virus infection occurs, and from CNS they may also spread to some other organs like the salivary glands, along the sensory and the autonomic nerves. So they may also spread to the other organs from brain to the to the other organs like salivary glands through sensory and autonomic nerves. That's why the saliva is also infective. This is the pathogenesis in simple terms. Now coming to the clinical manifestations of the rabies virus infections. Coming to the clinical manifestations. So in the clinical manifestation we have divided the clinical manifestation in three stages. The first stage is the prodromal stage, the second stage is the acute neurologic state, and the third stage is the coma and death stage. In the prodromal stage, there occurs fever, malaise, nausea, and vomiting. So these are the initial symptoms. These are the initial symptoms which are seen in the prodromal stage. Next comes the acute neurologic stage. So this stage sets when the furious rabies occurs. What is furious rabies? Furious rabies is characterized by the symptoms of the hyperexcitability, hydrophobia, aerophobia, etc. So when these signs appear, hyperexcitability, hydrophobia, aerophobia, then that is characterized as the furious rabies, and the stage is called as the acute neurologic stage. Acute neurologic stage. And after the acute neurologic stage there is a stage of coma and death, and once the coma occurs the death is inevitable and will occur within 14 days. After the coma, death is inevitable, will occur after the 14 days of coma. Now coming to the laboratory diagnosis of the rabies virus infection. So now coming to the laboratory diagnosis. Coming to the laboratory diagnosis of the rabies virus infection. So first step in the laboratory diagnosis is the specimen collection. We have to first collect the specimen for the diagnosis of the rabies virus. What are the specimens that we collect? The specimen that we collect are the corneal impression smear, hair follicles from the nape of neck, that means the hair follicles from the back of the neck, the saliva, and CSF and blood. These four samples are the most important samples, out of which this hair follicles from the nape of the neck is the most important sample for the diagnosis of the rabies virus infection. The second step is the rabies antigen detection. This is done with the hair follicles which are collected from the nape of the neck, and the method what we use for the rabies antigen detection is the direct immunofluorescence test, which is in short called as DIF, which is in short called as DIF test. Okay, direct immunofluorescence test. So by this test the antigen is detected. And how do we detect? We use monoclonal antibodies tagged with the fluorescent dye. When these monoclonal antibodies tagged with the fluorescent dye bind to the antigens, that leads to fluorescence, and that fluorescence is detected. As a result, the antigen is indirectly also detected. So that's how we detect the antigen rabies antigen. This test has very high sensitivity and very high specificity as well. That's why it is also the gold standard test for rabies diagnosis. And because of that, the hair follicles from the nape of the neck are the most important samples to be used. Are the most important samples to be used. Okay, out of all the samples, the hair follicles from the nape of the neck is most important because that is used for the rabies antigen detection. And since this rabies antigen detection is having high sensitivity and high specificity, that's why it is also the gold standard test for Rabies diagnosis is the gold standard test for rabies diagnosis. The third step in the diagnosis of rabies virus is the virus isolation. Now, this virus isolation, as we have read in the general virology, that is not done routinely. This is mainly for the research purposes, but for the sake of diagnosis of the rabies virus, we have to know this. So, in any virus isolation, we need some cell lines, and the preferred cell lines in case of the rabies virus detection are the mouse neuroblastoma cell line and the baby hamster kidney cell line. Mouse neuroblastoma cell line and the baby hamster kidney cell line. After choosing the cell lines, we have and after choosing the cell line and growing the virus over that cell line, we have to detect that virus. So the growth can be detected by using a specific monoclonal antibody tagged with the fluorescent dye, the same method what we have used in the rabies antigen detection, that is the DIF test, direct immunofluorescence test, is used to detect the antigens of the virus, and by that we can infer that the viruses has grown over the cell line. Other than that, we have always the PCR. The molecular methods are always there for detection of the virus over the cell lines. So by these two methods we can detect the virus over the cell lines. That is called as the virus isolation. The next step in diagnosis is the antibody detection. The next step is the antibody detection, and the antibody detection is basically done in the CSF and in blood. Antibody in CSF can be detected using a specific antigen. Again, the antibody is detected using the specific antigen of the rabies virus. Other than that, we have the molecular methods. So molecular methods are employed for the brain tissue and saliva. So for detection of the virus in the brain tissue and in saliva, we use the molecular methods, and in the molecular methods we use RT-PCR, because the virus is a RNA virus, so we have to detect the viral RNA, and for that we need RT-PCR. With the help of RT-PCR we detect the virus. Now the demonstration of the Negri bodies. Negri bodies is sometimes asked as short notes. Okay, so as a separate topic also, this is very important. The negative bodies are very important, and in the lab diagnosis also this point is very important. So demonstration of Negri bodies. Negri bodies demonstration are used for the post-mortem diagnosis of rabies. Are important for the post-mortem diagnosis of rabies. Now, what are those Negri bodies? What is the definition of the Negri bodies? So, Negri bodies are intracytoplasmic, eosinophilic, means somewhat red colored, round or oval inclusion bodies in the neurons, which are seen in case of the rabies virus infections. So, Negri bodies are intracytoplasmic, eosinophilic, round or oval inclusion bodies in the neurons seen in case of the rabies virus infection. Seen in case of the rabies virus infection. Now coming to the location of the location of these inclusion bodies. So inclusion bodies are commonly seen in the neurons of the cerebellum, hippocampus, and the brain stem. These sites are very important to remember, because these are the very important locations of the Negri bodies. Negri bodies are most commonly seen in the neurons of cerebellum, hippocampus, and the brain stem. What is the composition? What is the composition of these Negri bodies? These Negri bodies are formed by the aggregation of the viral proteins. They are formed by the aggregation of the viral protein. How do we demonstrate these Negri bodies? So for that we take the biopsy of the brain tissue and stain it with the H and E stain or the Sellers stain. We take the brain biopsy and we stain it with the H and E stain or the Sellers stain, and then we detect then we see the biopsy specimen under the light microscope. What do we see under the light microscope? So under the light microscope we'll see the intracytoplasmic eosinophilic inclusion bodies, which are nothing but the nacre bodies seen in the infection of the rabies virus. What can we interpret by the detection of the rabies, by the detection of the inclusion bodies in the neurons? So although these are the pathognomonic, these are the inclusion, these negative bodies are the pathognomonic for the rabies virus infection, but still sometimes, like in about 20% of cases, the negative bodies are not seen in the neurons. So a negative smear cannot completely exclude the diagnosis of the rabies. Okay, so this is all about the diagnosis of the rabies virus. What we have read is the specimen collection. So in which we have to collect four specimen: the corneal impression specimen, hair follicles from the nape of neck, saliva, CSF, and blood. The rabies antigen detection, that is the gold standard test for the rabies diagnosis. Then the virus isolation, which is done with by using two cell lines. Then antibody detection in the CSF, antibody detection in the CSF and blood. Then molecular method, that is the RT-PCR method we use. Then demonstration of the negri bodies, and interpretation after demonstration of the negri bodies. So this is all about the lab diagnosis of the rabies virus infection. We will see the prevention of the rabies virus in the next part of this video. So till then.